Hey, what's up everybody, and welcome back to the Sanctuary. I'm Professor C, and we're going to talk about A&P some more today. Specifically, I'm going to get into the concept of the cell cycle all the way through mitosis. So, let's get it. Let's talk about, how you as a life form have a life cycle, and you go through certain changes, through certain phases of your life. Well, cells also have the same thing. We call it the cell cycle. So let's look at it. From the time that a cell is formed to the time that it dies, all the changes that occur within that cell's life. Two major phases, but interphase and mitosis. Now, mitosis is easy to spot because it's really short. And it's right up here, and you can see it's further divided into things like prophase, metaphase, anaphase, and telophase. But there's mitosis, it's easy to see. But here's the term interphase, and it's actually all of this all the way around to here. So there's a better way to see it is most of the time a cell is in interphase, and then a little bit of time it goes into this multi-phase process called mitosis. So let's look at interphase first. Again, interphase is anything that's not mitosis. 90% of the time. Again, this picture is pretty good at showing a circle with most of it being eaten up, if you will, by interphase, and mitosis is a small part. So 90% of the time, a cell is an interphase. If you watched the prior videos about DNA, and the different forms that it takes inside of a human cell, this is a number we've seen before. Chromatin is found in the nucleus 90% of the time, and so during interphase, when we're using DNA to create proteins and do the things that cells do, we have DNA in its open, uncondensed chromatin state. Three major phases as you see here. Now G0, I put some stars around it because it's not shown, and I can draw it kind of like this. Let's say right here when you're born, there's a G0 phase. And I'm drawing it here because it takes us out of the cycle. So if you are a cell that goes into a G0 state, you are in a permanent state with no division. Now that's arguable depending on how far you want to jump in there. So cells that do this would be like skeletal muscle, heart tissue, cardiac muscle, brain tissue, nervous tissue. Generally, when these tissues, these cell types achieve maturity, there is no more division. They go into a G0 state. This is why it's not good to break your brain cells. It's not good to damage your skeletal muscle cells because they don't grow back at some point. We're going to try to talk about normal cells. So the first real phase is called G1. Now you think of G meaning growth. That's what I always did, but it actually refers to, make that a little bit more pretty, gap number 1. Again, I thought it meant growth 1, and it never stopped me 1 minute, not knowing that it was gap all the time. But what's going on here? Well, growth, metabolism, and growth, using energy, eating food, getting bigger. Right before we divide in a little bit through mitosis and so forth, we need to get bigger before we can divide, or we have a bunch of teeny tiny offspring. After G1 is finished, we enter this what's called an S phase, sometimes R. In this figure, I can circle it down here, this green kind of area. They're calling it S, but it could be R, depending on what text you're reading. S stands for synthesis of DNA. R would be the replication of DNA. Now, when we talk about DNA replication, we're going to use that term, replication. I prefer to use that term, but other books just call it S phase, right? So DNA is copied here. Remember, we're about to divide ourselves when we do mitosis. If we're going to divide ourselves, we don't want to give half of our DNA to our offspring. If we only give half the information, and then they give half to their offspring, and then they give half, pretty soon we're out of info. So what we want to do is take all of our info as a cell and double it, copy it all, so we can give half of it later, which is actually a full set. That's the whole point of the S phase or the R phase and interphase, is where we copy DNA. Now I'm going to remind us of this when we talk about DNA replication in the next couple of talks. Then you go into a G phase. You actually don't double immediately after that. You go into another gap 2, where you grow, metabolize, and you get fully prepared for division. Anywhere along the way, there are checkpoints, and if you fail one of these checkpoints, you could be told that you don't need to participate in cell division any further, and you may get a signal to do autolysis, where again the lysosomes will devour the cell from the inside out. After G2, we're going to jump into mitosis. So there's the three major phases of interphase: G1, S or R phase. I prefer R, other places prefer S, and then a G2 phase. Growth, double the DNA, and then more growth. Mitosis then comes. And if you're having a little trouble remembering all the divisions here, I'm just going to help you. You do a little mnemonic. You say I, interphase, past, prophase, right? M, metaphase, A, anaphase, T, telophase. We're going to go over these, but just remember, I passed my anatomy test. Very easy. I PMAT, right? I passed my anatomy test. So mitosis is actually not cell division, but often people call it this. Most tissues use mitosis to repair, like your skin. You're always creating new skin. Your skin is an organ, and so when you damage your skin, you get a cut or whatever, the cells will replace themselves, every 3-4 weeks, and you'll have a whole new set of tissue. Of course, your skin looks like your other skin cells because you want it to be clones of each other. If you're trying to repair skin, you want to have more skin take its place. This is the concept of cells dividing through mitosis. Remember that some cells go into a G0. Like I mentioned in the previous slide, nervous tissue, skeletal muscle, cardiac muscle, they're not going to do mitosis when they are a mature cell. Okay, here comes the PMAT. Remember, I passed my anatomy test. 4 phases of nuclear division, and wow, that is so important. Mitosis is nuclear division. It's actually not cell division. Cell division is afterwards. And what I'll do is I can put a little thing here, and we can call this little sliver here cytokinesis. That's the actual cell splitting. So before we split the cell, we must divide the nucleus, and that's what mitosis is specifically. Now it turns out cytokinesis will begin right back here in anaphase, and there's a little overlap, and that does make it a little confusing. But what we're going to do in most ways you talk about this is first you will divide the nucleus, then you will divide the cell. All right, phase by phase. P is first. The way that you see this on most exams is they'll give you some sort of description of an event that's occurring during this phase. Very rarely do you see pictures of it, but I've put pictures here so you see it. If you have chromosomes, well, that's a good sign. Because remember, most of the time, the cell is an interphase, and in interphase you don't have chromosomes, you have chromatin, and you don't see any kind of nuclear material whatsoever. So here we have some nice chromosomes. They almost look like they've been dropped from the ceiling and have splattered on the floor, and the chromosomes are kind of spread out. Very good picture of what we call prophase. So things that would happen here, major events, the nuclear envelope, the thing that surrounds the nucleus, the double membrane, and the nucleolus, any globs of RNA in the center will vanish from the cell. They'll disappear. They're going to reappear later, but we got to get the chromosomes, that is the genetic material, out of the nucleus. So the nuclear envelope evaporates. The chromosomes are now actual chromosomes, and they look like a propeller, like that, stuck to another propeller, like that. So they get this classic X shape. Now in the pictures you can't see them up here as X's, but imagine a lot of little X's all jumbled up, kind of like floating in a cereal bowl, and that's why you can't see them, because they're all jumbled up together. So what we see here is one X-shaped chromosome, and these are called chromatids. And you see that word there, chromatids. So DNA condenses here into chromosomes, and if you could see them clearly, each would look like these double propeller structures, these X shapes. 2 chromatids join in the center by this little sticky ball right in the middle. This is called a centromere. Centrioles. Now we talked about a centrosome, and we talked about those 2 pasta-shaped guys called centrioles, and how they usually hung out together in the center of a cell at a 90-degree angle. Well, now what we're going to see is the centrioles will split apart. Fast forward a little bit, and you get something that looks like this classic metaphase. Now let me show you what you're seeing here. Here is the centriole. Here is a centriole. Remember, they used to be together at 90-degree angle. Now they've split apart. They're moving apart. In the center, globbed up in the middle, chromosomes are all in the center of the cell now, what's called the metaphase plate. And that's going to be the distinguishing feature. It says down here in the text, centromeres of the chromosomes will align at the equator, if you want, or the metaphase plate, kind of this imaginary line coming right down the middle of a cell. You see also these spindle fibers, these spider webs being shot out from the centrioles. They kind of look like this. They're reaching out and grabbing on to the centrioles, they're grabbing onto the centromeres of the chromosomes. And this is a very classic shape for metaphase. Let me get rid of the ink so you can see it better. Wonderful picture of metaphase. Spindle fibers are attached. At some point, if these centromeres keep pulling apart, someone's going to be ripped in half, and that's going to happen in the next phase. So here comes anaphase, right? P-M-A-T. Ana means away or apart. We saw that with kind of anatomy, to cut away or to cut apart, on our very first slide a long time ago. So again, just to refresh our memory, we've got a centriole here. It's moving this way. We've got a centriole here. It's moving this way. We have spindle fibers attached to our chromosomes. Same thing here. We've got spindle fibers, but now you can see what's happened. We've ripped those chromosomes apart. And so the chromosomes now just look like single propellers. It's hard to see that. This one's kind of folded over like that, so you can kind of get a better picture of what you're looking at. But this is anaphase. It's the shortest phase of the cell cycle. The centrioles, as they drift apart, continue to pull and pull the centromeres until we get a pop, a split, a rift, a rip, and the chromatids are now separated. Once the chromatids are separated, we call them chromosomes, and they will continue to be pulled toward the poles of the cell. Anaphase. We're going to finish up the job here with telo. I know a lot of people say telophase, but that's a different root, tela. So I'm going to say telo, and I'm going to stress the spelling there, telophase. Telo means tail, so this is the tail phase, meaning the end, the tail end of mitosis. And very clearly here, what we need to do is we need to get normal again, right? We need the cell to become a cell again. We need to recreate all the things that we destroyed and basically turn back into normal cell, even though there's two of them now, right? So we got to get back to interphase soon. We're going to leave this telophase. We're going to divide completely through cytokinesis, and then we're going to be in G1 again, so we have to get normal. Chromosomes must uncoil, uncondense back to their very opened chromatin state, which we'll need to use in G1 coming right up. New nuclear envelope has to form. The nucleoli have to reappear because we're about to enter interphase right after this. Now I know there's still the last bit of business we have to do here in the center. We still have to rip the cell apart. We've got the nucleus divided. Good. We made it through PMAT of mitosis. We've got the nucleus divided. Now let's divide the cell. Okay, cytokinesis. Again, if I were to add it to the picture, I would come over here again and I would put it right about here before we actually went into G1 again. So cytokinesis, after the nucleus is divided through mitosis, we do the final little bit. What you see here forming is, it looks like cleavage, right? It looks like we hit it with a cleaver. We took an axe and chopped it. So sometimes this is referred to as cleavage or a cleavage furrow. Looks like it's being choked here by some sort of belt, like we're constricting the belt tighter and tighter and tighter. And it looks like some point, if we keep constricting it, it will pop off altogether and separate completely, and we'll have two new cells. So during late anaphase, again, I said cytokinesis begins somewhere over here, and it continues through the final stages of telophase, where we finally finish dividing the nucleus. We'll get this cleavage furrow to appear, and then the two daughter cells, is what they're often called, although I hesitate to use that term a lot, are pinched apart. Each have an identical nucleus, and for all intents and purposes, these cells should be identical. They should be clones of one another, and that is the whole point. If you're trying to get skin cell to divide and become more skin cells, you don't want it to become an eyeball cell or a liver cell. You want it to become another skin cell. Well, what about getting old? And then finally, what about, passing on, what we call senescence? This has a lot to do with this concept of the cell cycle. In fact, it has a lot to do with this concept of something called the Hayflick limit of mitosis. There is a limit to the number of times the cell can do interphase mitosis, interphase mitosis, interphase mitosis. Problems happen. Every time you do this, you do a little bit of damage, right? You pass on errors, you pass on mistakes to the new cells. Just like if you were to take a piece of paper in a copy machine and keep copying the copy and copying the copy, eventually you run out of info. It starts to deteriorate. And this seems to be one of the main reasons why cells get old, people get old, and they eventually pass away. So what's been discovered is 50 times is about that limit. And we know this for these reasons. So when we look at a chromosome, and I'll kind of just draw one here, kind of sketched out that propeller shape. On the ends of the chromosome, there's some special caps that keep this chromosome from unraveling. And these are called telo. We've seen that root tail mere thingy unit, right? We've seen that too. Telomere tail thingy. There are telomeres at the end of your chromosomes. And every time you do a replication, a division, these telomeres get shorter and shorter and shorter. And after about 40 to 60 times, somewhere about 50, the telomeres will unravel completely, and the chromosome will not hold together, and that cell will be told to do autolysis, to destroy itself. What if, though, what if there were a way of rebuilding these telomeres? What if there were an enzyme out there? Maybe one called, I don't know, telomerase, that would help rebuild these telomeres. Could we extend the length of the Hayflick limit? Could we keep cells living longer? Could we keep people living longer? Well, I'd say if you're interested in it, do some research on this. It's very interesting. It looks like with the right science, we could get humans to live anywhere from 125 years to perhaps even 400 years old, which is bizarre and really set for another talk on another day. But there is some limit to the number of times a cell can crank through the cell cycle before it is told you have too much damage and you need to end yourself. Okay, as usual, thanks for watching the video. If you want, check out more in the series. See you for the next one. Bye-bye.